You've learned at this point that scientific research is about asking questions and collecting data to address them. That data needs to be analyzed in order to make sense of it. Let's talk a little bit more about the data we collect and how we can understand what it means. In developing your research plan, you had to make a very important decision, whether you were going to conduct experimental research or observational research. In experimental research, a scientist will take samples that are all the same at the start, then assign them to groups that will be treated differently. They then analyze to see what happens to the groups. In observational research, a scientist will sort their samples based on a characteristic they already have, then treat all samples the same way. They'll watch to see how the groups respond. It's important to know which category your plan falls into because it will affect the way you analyze and discuss the data. You'll remember that the variables we change are called independent variables, and the variables we're testing are called dependent variables. Every study can have more than one of each type, but introducing too many independent variables can make it hard to determine which one caused the changes we see. So what is a dataset, and how can we talk about it? A dataset is a collection of raw information that describes a particular characteristic for the group of samples or subjects. Data can be quantitative or qualitative. Quantitative data is numerical and thus easier to run statistics and analysis on. Qualitative data is descriptive. Usually we can assign it values in order to better analyze it. There are a number of characteristics that we'll want to know about when we talk about a dataset. The first way to talk about a dataset is to discuss its central tendency. This includes talking about things like the mean or median, which tells us where the middle of the data lies. These terms are probably familiar to you. The mean is the average of all the data points in the set, while the median is a center value in an ordered list. Another way to characterize a dataset is by its distribution. The distribution gives us information about how spread out the data is. One way to measure it is by calculating the standard deviation. This is a measure of how far away each data point is from the mean, on average. A larger standard deviation means our data is more spread out. There are many ways to determine distribution, but a good way to visualize it is through a frequency histogram. To create one, we'll plot recorded measurements on the x-axis and how many samples reported each measurement, or the frequency, on the y. If the measurements with the highest frequency occur in the middle, close to the median and mean, our data may be considered to be normally distributed. Normally distributed data has all measures of central tendency fall in the same place, with roughly 68% of the data falling within one standard deviation of the mean, and 95% falling within two standard deviations. It's important to note that just because a certain characteristic is normally distributed within a population does not mean it will be normally distributed for every sample set taken from that population. A population encompasses every member of a group and is often too large to test in entirety, so we collect data from a small section of it, called a sample. The sample size is another important characteristic of a dataset that we'll need to know and understand in order to analyze it. The size of the sample can change for different experiments, but needs to be large enough to be representative of the population as a whole. With the right sample size, we'll be able to draw conclusions about a larger population, based on the experiments we run on our sample. If a sample size is too large, it may be too time-consuming or expensive to test. But if it's too small, we can't be sure that it correctly characterizes the entire population, and therefore we can't determine if our results are significant. We can determine significance by running statistical tests on our dataset. There are many different types of tests that will serve different purposes, but in general they use the distribution and central tendency of the data from two groups to determine whether they differ in an important way. Often these groups are an experimental and a control group, but you can also test between two experimental or two control groups. To say that results were statistically significant means we determined that two groups differed substantially in their reported values, and the chance this occurred by accident was lower than a predetermined threshold. The most common way we set this threshold is by determining a p-value. The p-value is a maximum allowable probability that the results occurred in error, so the lower it is, the more sure we are of our results. A common p-value in science is 0.05, which means there is a 5% chance that variations between two data sets occurred by error. This allows us to be 95% sure that the differences were related to the variables we were testing. For each test we run, we'll compare the p-value calculated from the data to the threshold p-value. If the calculated value is below our threshold, then we can confidently say that any apparent differences between the two groups were caused by the variables we changed and not due to error. This is why it's important to carry out experimental designs carefully. Any unintentional differences between the way groups of samples are treated could lead to incorrect results when determining significance. When you're reading a scientific paper, you may not always have knowledge of the specific statistical tests a researcher used, but now you should have a better understanding of what they mean when discussing the significance of their results.